Thanks a lot, Manuel, for the nice introduction, and I'm very grateful for the invitation to be here in Oxford again. I think the last time I was here was 20 years ago at ECVP in Oxford, and that was a particularly wonderful location for me because it was really the first time I was invited to give a keynote lecture at sort of a substantial international meeting. I was very happy about that, and I'm very glad to be here again. I've worked on visual perception, especially color vision, for a really long time, for more than 30 years now. And it was about 25 years ago that I sat in front of an eye tracker for the first time, and I was immediately fascinated by the richness of the data that you could obtain by studying eye movements. But for the longest period of time, these two research interests, visual perception and eye movements, have basically run along in parallel with not that many interactions between them, and that has only changed recently. And I will present to you today some experiments that really led me to the belief that we can only fully understand visual perception and the oculomotor system if we study both the eye movements and perception at the same time. I'll give you some examples showing you that basically the way we move the eyes around really optimizes our perception of the world, for example, judgments of how bright an object is or where a stimulus is headed to. And at the same time, these judgments are affected by the way we move our eyes. So what we see depends on the way we move our eyes, and how we move our eyes depends on what we need to see. And this is really quite distinct from 90% of the approaches that in visual perception and in the oculomotor system, where you really get the impression that eye movements and visual perception are enemies of each other. And the one ingredient that's so typical for that approach, and that most papers have in common, is this little spot here. It's the fixation spot that basically in visual perception experiments makes sure that the observers don't move their eyes. And in experiments on eye movements, mostly the only visual target is this little spot to basically have a minimum visual stimulus that you need to study the eye movements. So today I will hope to convince you that we should really put both of these systems together. But before I do so, I'd like to make a little footnote on the fixation spot here. It's already on the title slide, which means that nobody has really studied very much the fixation spot or what its properties are. And in most papers, it just says, well, we had a fixation spot. And then we actually went together with Lori Thaler, who's now a lecturer in Durham. We looked at all these papers, and there was just a big mix of just about anything that was used as a fixation spot, if people wrote it in their paper at all. So we actually went and tried out many different fixation spots and measured basically which one showed, for which one the eyes showed the least amount of dispersion and the smallest number of microsaccades. And this one here is the winner. It's just a superposition of 3 targets. So if you need a really good static fixation spot, this is what you should use. And of course, I mean, the reason why the fixation spot is so popular is, of course, because our retina is not homogeneous, and we want to control where the information is presented. And this is illustrated here in this distribution of the cone photoreceptors across the retina. And most of the time, basically, we want to stimulate where the eye sees best. I mean, the fovea, where the density of cone photoreceptors is basically 100 times as high as in the peripheral retina. And you can see here already that the foveal region is relatively small. It's just about the size of a thumbnail at arm's length, where we really see the world in sharp detail. And it wouldn't do us any good to have such a small fovea if there weren't these muscles attached to the eyeball that allow you to reposition the fovea. Just this slide illustrates a bit more how small the fovea is now. This is the foveal region in the visual field in both eyes. And if you sort of put a map of the world on top of that, if you see the eyeball as the whole Earth, then basically you can see that the fovea is about the size of Ireland on this map. So it's really small, and the Germany is about the size of the blind spot. It's bigger, but so, and in order to help us see the whole world in sharp detail, we have mainly two different kinds of eye movements that allow us to travel around, namely saccades to sort of make big steps, and pursued eye movements to make sort of slowly follow moving objects. And of course, the major benefits of these eye movements has been studied for a very long time, and they are very clear. For saccades, which are illustrated here, they of course improve the acuity of the object that we're foveating, and we can move the eye acuity around. The disadvantages are also quite obvious. During the saccades, when the eye moves at a very high speed, we're pretty much insensitive to visual stimulation. And of course, the saccades dissociate the object and the retinal position, and that's important because the visual cortex is organized in a retinotopic way. Basically, all the receptive fields in visual cortex get a new input from one saccade to the other. Pursued eye movements, it's basically similar, just for motion. It of course improves the acuity for moving objects, because the moving object is always in the fovea. But of course, it at the same time blurs the stationary background, the background that's stationary in the world, but then moving on the retina. And it dissociates object and retinal motion. So if you move your eye, you move your eyes, the object is stationary, then it moves across the retina, and somehow this has to be disentangled. But as I said, basically these obvious effects have been studied quite a lot. And in my talk, I would like to emphasize that there's more than these very general aspects, that eye movement and task interact very closely to achieve an optimal performance of many perceptual tasks. Now, of course, it's known that eye movements and task interact very closely from the literature, where people like Mike Land or Mary Hago and Dana Ballard have studied the way we move our eyes when we do an actual motor task. For example, Mike Land when studying making tea, or Mary Hago and Dana Ballard, they watched the eye movements of the observers when they were making peanut butter and jelly sandwiches. So, and in this case, basically, the eyes always go to the places that are next really needed, where information is needed for the task at hand. So in this case, the eyes always make the required information available for perception that can then guide our action system. And I'll show you just a brief example from my own work that's even much more basic than making tea. I mean, it's just by grasping an object. So in our experiment, we had 7 different objects shown here, and they were mounted on a screen on a plexiglass frame. So those were real 3-dimensional objects. And the observer was fixating here, and then they had 2 different tasks. In one set of trials, they just made a saccade from this fixation region over there to the object. And in the other set of trials, they had to grasp the object with a precision grip, with a thumb and index finger. And you can see an example trial here. So green is the gaze trajectory, and the black are the trajectories of the thumb that sort of touches the object triangle here at the base, and the index finger, which contacts the triangular shape here at the tip. And that was actually a very typical experiment for the grasping trials. In the viewing trials, what we observed, this shown here on the left, was mainly that the observers made a saccade to the center of the object, the center of gravity, or here the intersection of these bars, and that's where that's very well known from the oculomotor literature. You always basically go to the center of gravity of the objects when you first saccade there. In the grasping trials, we also observed that observers made more saccades or saccades in addition or instead to hear the top contact points of the object where the fingers will contact, and basically there was always the contact position of the index finger. Now, of course, you can argue, well, it's not really the index finger, it's just because it's on top, but we did control experiments where we rotated the objects and forced the observers to grasp the object in different ways, and basically observers always made a saccade to the future contact location of the index finger, not the thumb, and presumably because there's more flexibility in the movement of the index finger. And interestingly, there are a lot of people out there who didn't believe us when we first came out with this, and they all redid these experiments, and they all found the same thing. So that was quite reassuring. So that's just an example, really, how deeply ingrained this eye movement analysis are in any sort of motor task, even sort of this very, very basic task of grasping an object. The eye movements are very important whenever we do something. And in what it follows, I will show you that they're also essential for the way we sample the world for particular perceptual judgments. I would wonder why do we care that much about perceptual judgments, or particularly one, or purely perceptual task? Well, there is actually one example for a perceptual task where the eye movement contributions have been studied a lot. And that's basically what we as scientists do most of the day, and I mean just reading. So when you read, you pick up you only have sort of 4 or 5 letters available that you can see that you can really recognize the letters, and that's why you basically have to go from word to word. And the eye movements for reading are actually really optimized quite well. So you skip words that are highly predictable, and sometimes when something is very complicated, then you might actually have to make a saccade backward. And it's really the eye movements in this task are really perfectly matched to the statistics of the text and the difficulty of the information. I will show you now that the way we sample the world through the choice of fixations that we make with the saccades really massively influences a very basic perceptual task, namely lightness judgments. And then I'll also show you that saccades are used to calibrate the foveal and the peripheral visual field to achieve perceptual stability. And this slide from Eli Brenner's lab really illustrates very nicely what I mean by sampling. I mean, when you look at it, you obviously see a yellow grapefruit and a green apple. But if you look at it more closely, you can see, as illustrated here in the sidebar, that sort of the color on the surface of the grapefruit and on the apple actually vary quite a bit, and there's sort of regions in there that have quite distinct colors, and some of them actually identical between the two objects. Others are very, like the yellow here is very typical for the object, and the green here for the apple. And there's a whole range of reasons why color varies here. It has to do with pigmentation, with shading, and illumination changes as well. So the question is how we arrive from this whole distribution of colors to sort of the judgment, our estimate that of the object's color, namely that the grapefruit is yellow and that the apple is green. And I will show you that the process, how we sample this object with various fixation, really does play a major role for how the object appears. Now, this is the task that we used, and this is basically it's the most simple experiment you can think of. So we just took a series of objects that happened to be around in the lab, put them on a black pedestal in front of a screen, and on the screen we displayed a little patch of light. And the task of the observer was simply to adjust this patch of light so that it would match the color of the object. And at the same time, we recorded the fixations of the observer, and then, of course, and we took spectral radiometric measurements of the object so that we would know the ground truth of what the spectral reflectance at these various points really is. And the observers could adjust this point sort of in the saturation, in the hue, and with respect to the lightness. And two things. First of all, observers really did this task perfectly for hue. And that's not very surprising because we're very sensitive to hue differences, and they adjusted the hue of this patch to perfectly match the hue of the object. So that nothing special to report here. For the saturation, we realized after we did the experiments that we didn't really know what saturation was. And after studying the literature on saturation, which pretty much doesn't exist, we realized that, well, it's really undefined. I mean, many people even define saturation as what's left over once you've dealt with intensity and hue. And so we started a whole research program trying to work out how humans perceive saturation, but basically for the beginning, we didn't really further look at the saturation data here. What I will present to you today was the lightness aspects. That is how observers estimated the intensity of these objects, and those turned out to be really very interesting. The most important result of all is really that observers can do the task. So they don't ask, well, what do you mean by adjusting this patch? Should I give it the same color as here, this bright region, or here, this dark region, or what should I do? They adjusted this patch to match sort of the invariant object properties, the reflecting properties of the object, and that's I think that's a very important result. And here is illustrated how they might sample the object with their eyes, and then they go to adjust to the bar, and that's how they basically performed the task. This shows the results for this orange paper cone here, and what's plotted on the y-axis is basically an estimate of the luminance that the observer adjusted here, expressed as L star, which is sort of a particular optical society measure. It's called lightness, but it's, well, I shouldn't have started that point. But you can think of it as luminance. And this shows the mean luminance of all the pixels in the paper cone. This shows plus or minus 1 standard deviation, and the thin vertical line shows the range of luminances in the paper cone. So it goes really from very dark to relatively bright. These points here show the matches of the observer, and the cross being the mean. And what you can see here is the mean of the matches, that what the observers adjust as the luminance or the lightness is very different from the mean luminance of the object. So that's very clear. Instead, what they adjust is very similar to these three example patches here, which is basically, one is the maximum lightness within the object. This here is the actual match, and a little bit above here is basically the limit, what the lightest that the object could possibly be, maybe when it's oriented perpendicular to the light source. So it's very clear that what the observers match is close to the luminance of the brightest parts within the object, and it's close to this upper limit, and they do not use the mean luminance as their estimate. Now, why do observers do this? Why do they use this strange strategy? It's strange from the viewpoint of statistics. If you learn statistics, well, you hear all the time the mean is great, the maximum is really bad. It's unstable, it changes the more, it's just bad. So in this case, observers use the maximum. And so we wanted to know why and how do they get there. And it turns out that if you look at the fixations that the observers make, that those are also centered in this task on the brightest parts. So here, this graph divides up the range of luminances into 9 different bins, and it plots how frequent the image regions having a certain luminance were fixated. And what you can see here is that those regions having a luminance that's very similar to what was matched by the observers, namely the brighter parts, were nearly always fixated, and those that are very different from the match, the dark ones, were hardly ever fixated. So we do match the brighter parts, and we look at the brighter parts, and what does that mean? I mean, it's a correlation. So does the perception drive the eye movements, or do the eye movements affect our perception? So it would be nice to actually be able to use some experimental approach to that, where we manipulate something, either the eye movements or perception. So we figured that we could manipulate the eye movements of the observer, and the way we did that was using a gaze contingent paradigm. So in this case, we force the observers to look at a certain part of the object, either a bright part or a dark part. And the way we can do this is by telling them where to look, by displaying this red spot here. And if as long as they fixate here in this spot, which we measure with our eye tracker, we display the whole object here. If they go away from the spot, the object magically disappears, and they only see the red spot. The red spot is not there when they fixate there. The red spot appears, and they know where they have to make a saccade to for the object to reappear. And then if they go to other points within the object, also this disappears. But the only thing is, when they're on the matching disk, they can see the object in peripheral view. That's how these experiments work. And of course, I wouldn't show you this if the results didn't come out in the interesting way. The interesting way being that when observers are forced to fixate a dark region of the object, indicated here, then they adjust the lightness, the matching lightness, to be darker than when they fixate on a brighter region. That is, the way they perceive the lightness of the object depends on where on the object they fixate. And of course, there's many different control experiments here, just making sure that observers do not sort of make a local luminance match. It's not that they replicate the luminance of the object here and there. In that case, the effect would be much, much bigger. And we also used other tasks that really get the observers to make lightness judgments, like, for example, picking a piece of paper to match the object. But all of that basically the results were all the same, and it's really the lightness that's affected, the perceived lightness that's affected on where the observers fixate. So they produce estimates of global lightness. They're close to the maximum. They fixate these points, and they match a higher lightness when forced to fixate a bright region of the image. And that basically the summary of this is that the way we perceive the lightness of objects is driven by how we look at them. And the strange thing is I mean, that doesn't sound very positive, right? I mean, like if you would have an object and you would rotate it around, then, I mean, you don't want it to change its brightness. It's still the same object, so it shouldn't change. We want lightness constancy is what we want. And what I'm going to show you now quickly is that this strategy of looking at the brightest region actually is something that supports lightness constancy, that the maximum is basically the best estimate that we can make of these objects with respect to lightness constancy. And the way we looked at this was by doing some simulations where we used computer graphics to generate many different views, 400 views of many different objects illustrated by natural light fields. So those are just 3D models on a computer. We illuminate them by a natural light field, and then we can basically calculate the intensity image that would get projected into the observer's eyes. And then we want to figure out which statistic, which estimate of the lightness of the object using these intensity samples for each object under the 400 different views is the most stable, that is, doesn't change. So that's basically the reliability. Basically just gives you tells you how stable these estimates are under changes of the scene geometry. And the statistics that we used, of course, one was the mean and the other ones were the 100 percentiles of the intensity distribution. So you calculate the variance over these 400 different views for each one of these 101 statistics. And you can see that for the mean, we get this relatively high value, and then we get very small standard deviation, that is very high reliability for the minimum and for the maximum. And you can say, well, the minimum works really best. But the minimum is basically, that's amounting to saying that everything's black. So it might be best, but it's not very informative. And of course, even here with the maximum, the reliability is only one aspect. You can be extremely reliable, but at the same time always be wrong, right? So like if you have a scale that no matter how much different people weigh always gives the same output, then no matter, I mean, if the same person gets on it, gives the same output, it's extremely reliable, but it's still broken. So you need another estimate, and that's the validity. Basically, how well can this estimate discriminate between different lightnesses? And there it's clear that the maximum is here really the best, achieving a very high level of diagnosticity. And so the maximum is really the best estimate in this lightness task, and that seems to be what the observers are using, and that is supported by the strategy of how we look at these objects. And then, of course, there's much more to lightness than just making these estimates of isolated objects. There's like tons of visual illusions that are well known, and we looked at a few of them, and basically in all the cases what we found was that the fixation pattern was really supportive of that particular estimate that worked best in that situation. So basically the perception guides the eyes, and then the chosen fixations positions sort of amplify the chosen perceptual solution. I mean, you still perceive the world if you just fixate, but the eye movements, they improve upon this estimate. And sampling the world in a manner suitable for a given perceptual task seems to be a crucial contribution of eye movements to perception. Now, in the next much shorter part, I will still talk about saccades and fixations, and I will show you how saccades are used to calibrate the different parts of the visual field. And this calibration is very important because when you look at the world should appear stable. That is, if you have an object in peripheral viewing and then you make a saccade to it, it doesn't usually change its color or size or anything. And somehow we need to learn this association between the different parts of the visual field, because the visual system in the fovea and periphery can actually be quite distinct. So if you fixate here on the grapefruit, the sensitivity of the color vision system actually decreases in the periphery in a differential way for red, green, and blue, yellow, and luminance. So if all of that went through without calibration, then the perceived colors might actually change for the object as you look around in the world, which obviously it doesn't. And with size, it's even more dramatic, because if an object gets projected into, here's a sort of a very simplified representation of V1, and here you have the apple in foveal view. It basically takes up a lot of cortical space, and if you compared to the grapefruit, and if you look at it in peripheral view, then the grapefruit here is very tiny. So we need to an efficient way to learn this relationship between foveal and peripheral view. And it has been suggested by O'Regan and Alvanoe in 2001, and actually by Helmholtz in 1867, that basically it's the saccades that associate the peripheral effects of a stimulus with the subsequent foveal effects. And basically our visual system just learns this contiguity, and that then basically our peripheral perception, what we perceive in the periphery, is just basically a prediction of the foveal effects. So we learn what will come next when we foveally view the object, and then we use this prediction as our estimate. But that idea has never really been tested strongly. So what we did an experiment where we used a gaze contingent display, and the observer simply had to compare the size of a foveal and a peripherally viewed object. Just say which one of these disks is bigger, and then make a saccade to the other disk, and then make a second task to judge the orientation of a small protrusion here that couldn't be seen in peripheral view, but only in foveal view after the saccade, and that was just there so that observers would really pay attention to this secondary task. And of course, the crucial manipulation is that while the observers make the saccade, we change the size of this other object slightly. Either make it 10% bigger or 10% smaller, or in a control group, we didn't have any size change. And all of this goes unnoticed because we're not very sensitive to these changes of the world during a saccade. And we actually did some experiments where we measured whether people could notice this, and this was all really way below threshold. And here are the results. So this went on for 400 trials, or 500, and here's on the y-axis is basically the perceived size of the peripheral object. And this is the baseline, and you can see that in the baseline already the peripheral object appears smaller than the foveally viewed object. In the beginning we were quite excited about that, but then we realized that many other people before us had already found this, and that it depends on what conditions exactly are used, so it's not that interesting of a result. But the interesting result was when we started to change the size of the peripherally viewed object, and that was, so we had 100 trials during which everything was stable, nothing changed size, and then we gradually introduced this 10% size change, and then for the next 250 trials it was that change was stable. So we really wanted to make sure that observers don't notice anything is funny with this display. And the black curve here, that shows the control group, that's relatively stable, nothing much changes. The red experimental group, the peripheral size got smaller, and then they estimate that the observers make before executing the saccade, right? Just in this, before they do anything, actually becomes lower, and for the group where the size increases, it becomes bigger here. So there is a quite massive effect here. And then when observers came back on the next day, when they started out again, it was interesting that the effect And on the next day, we changed the assignments of the group. So the ones that had the decrease on the first day got the increase on the second day, and the other way around. And of course, the results then reversed. But interestingly, the effect here on the start of the second day was there. You would think that if we calibrate the saccades of these observers, then they would go home and make many more saccades, and basically this would wash out, surely wash out after a while, which it doesn't seem to do. But there's two possible reasons for that. One is that this size learning is very specific to the particular display. Like if you sit in front of a computer and do this experiment, which didn't seem very plausible. But the other thing that is, I mean, these saccades that people make are relatively large, 20 degree saccades exactly to the right. So we gave people mobile eye trackers and had them, they were wearing them for hours on, and basically it turns out that they hardly ever make a saccade that's 20 that's that large, 20 degrees to the right. Most in the real world, the saccades are 5 degrees and less. So that's why this doesn't wash out, because they don't have the opportunity in the real world to relearn things. And that's also gives already the result of the other experiment, that this is relatively specific to a particular saccade size. It doesn't generalize to the whole visual field. Initially we had thought, well, maybe it's this M factor, this magnification factor that people are learning, but it really is very specific to a particular saccade. But it's only a few hundred trials are enough to induce this recalibration. And yeah, it's local, but surprisingly it did generalize to the opposite hemifield. But that shows that saccades are really very important for not only for sampling, but also for this calibration, so that the visual world really appears stable in the whole visual field when we make saccades. Now in the second, much shorter part of the presentation, I will look at the other kind of eye movements that we make a lot, namely pursuit. And pursuit eye movements are interesting because we really need a visual motion stimulus to make this pursuit. We cannot make them in the absence of motion. But they're also interesting because when you look at what's salient in the world, and not in the laboratory where things would come out of nowhere and then of course you would look there, or where things would flicker, but if you look around in the real world, and we did that with colleagues in Lübeck in computer scientists, we really created these natural videos, and the little red spots here are the fixation positions of 50 different observers. And you can see that most of the time people are all over the place, indicating that if you view these static scenes, it's very difficult to predict where observers would fixate. And the green bars here indicate the consistency of the fixation of the different observers. So if the bars are here, then they're all in different places. If the bars are up here, then everybody's looking at the same thing. And you can see that when one of the ducks here starts to fly, then everybody's going there and following the duck. So for saccades, these motion onsets are highly salient events. And what people usually do is they go there with a saccade, and then they follow the object with smooth pursuit. So that's why smooth saccades and smooth pursuit really work together very closely. The one nice thing about this video, which we didn't do on purpose, but it just we realized later, if you let one of the salience algorithms run on these video, then, or on the image, then basically the most salient object is this big red spot here, and nobody ever looks at it. So what I want to show you now for pursuit is that basically also when we execute pursuit, that it does have an effect on how we see the world, on visual perception. And here, the very basic thing is actually visual sensitivity. So when we started these experiments, we wanted just to do something similar to what's known for saccadic suppression. That is, we basically had observers follow a moving target, and then we presented flashes either on the top or on the bottom, and observers just had to say whether it was on top and bottom, and we wanted to see how that compares to a fixation condition. And when we did this experiment for luminant stimuli, that is just a white or a black flash, then we found that people were about 5% worse during pursuit. So here sensitivity during fixation is higher than sensitivity during pursuit, and the reason being mainly probably because people pay attention to the pursuit target, and they lose those resources for detecting the slightly peripheral stimulus. But the amazing thing that we found was that when you do this with color for a red or a green flash, then people actually get better. So there's really an improvement there of about 15%. And that's a bit strange because kind of color and motion really seem to be quite independent of each other. And here still you get this improvement when you pursue an object. So there were, at first we thought that, well, it's possible that this could be a byproduct of the rotation of the eyes. But there's two reasons. When you study the vestibulo-ocular reflex, where basically you fixate and you move your head, you get the same eye rotations, but there is no effect on sensitivity. And the other thing was that when we studied the dynamics of the effect during the initiation of smooth pursuit, here this red curve shows the eye velocity. In the sort of typical rash-based paradigm, the eye takes off and then reaches steady state. And when you measure sort of brief flashes, either before, when the eye is still stationary, or when it takes off, you can see that the improvement in performance, so it basically goes up from 60% here to 90% here, but this improvement comes about 50 milliseconds before the eye starts to move. So this is really seems to be an active change where the visual system sort of fiddles with some knobs, and at the same time that the signal gets sent to the eye muscles to start moving, it basically tells the early visual regions that it should readjust the various sensitivities. And now the question is, well, why would it adjust the sensitivity so that you better see color? There's one explanation having to do with color would be that basically that would prevent the background from blurring as much as it may, and color is mostly affected because you're not very sensitive to color at high temporal frequencies. The alternative is actually be that, and I think that's my favorite hypothesis, that it adjusts the part of the visual system that has a high sensitivity to small changes in speed, to low speeds, because when the stimulus is stationary and you want to pursue it as well as you can, then you want to be sensitive to small deviations in speed in order to correct the pursuit. And it's known from the oculomotor literature that there are changes like that going on. And I think what's happening in the visual system is that the parvocellular system gets a little bit turned up, and that happens to be also the part of the visual system that's sensitive to color. So color would only be a side effect here. But it's clear that there are these dynamic changes going on during pursuit initiation. What other improvements could there be during pursuit? Well, if you want to know, you have to know the speed of the object, but you also want to know the direction the objects are going. So we also measured the directional sensitivity to either during fixation or during pursuit. And in this case, the observers were fixating, then this is a paradigm that I call ice soccer, because you basically have this ball here, and then it starts moving towards the goal, and basically the observer has to say whether the ball will hit the goal or miss the goal. And then this is the pursuit condition, where the observers actually initiate pursuit, and there's the control condition, where basically the goal comes towards the ball, so that the retinal stimulation would be as similar as possible. And what we found in this paradigm was really that here's three different presentation durations, the fixation condition and the pursuit condition. You can see that this d prime here, performance in the pursuit, is always better than during the fixation. And the same holds for a more difficult version of the task, where the distance between the ball and the goal is higher. Still, pursuit is better. During pursuit, you're better than during fixation. And I think this is interesting, because what it tells you is that if you want to judge the direction of an object, of a moving object, that you should pursue it. And that's basically what sports coaches have been telling the players all along. Just keep your eyes on the ball, and that seems to be sort of a very good strategy in these cases. There's still the question of how this is happening and why it's happening. One possibility is that basically the efference copy signal gives you another signal that can be used to analyze the direction, and by combining this with the error signal, you get better performance. But we actually think that it's more of an attentional effect. We did experiments. I'm just really going to go over this very briefly and then stop. Where we had observers pursue a random dot field, and then during pursuit, we injected a small perturbation of 20% of the dots. 20% of the dots moving in this sector here sort of were moving in a slightly different direction during pursuit. So it could be upward or downward or whatever. And when we did that, we found the results are shown here. Show here the angle of the perturbation. So in this case, 90 degrees, the 20% of the dots would go upward, and 0 is of course no perturbation. And this is the change in the eye direction. And what you can see here is, surprisingly, that in the cases where the vertical component is actually biggest, like here, there's hardly any effect on the eye. So basically, when the eye is going horizontal and you add some perturbation in the vertical, the eye just ignores that. And it's only sort of the smaller deviations, like here 30 and 15 degrees, that get the eyes to change around. So it seems that the oculomotor system has some assumption of inertia built in, that prevents it from changing the direction very rapidly. And actually, the different curves here are at different times, and you can see that the angle with the maximum perturbation really becomes smaller and smaller as time goes on. That's indicated here. And if you correct all these curves for the vertical component, which is of course much bigger here for 90 degrees than it is for 30 or 15 degrees, then you actually get these tuning curves, and basically it shows you the longer pursuit goes on, the narrower the directional tuning becomes. And it's this narrower directional tuning that improves the judgments for the directional selectivity. At least that's what I would assume. So in summary, basically which I showed you, that we sample the world in an optimal way for lightness perception. We use these eye movements to achieve calibration across the visual field, and that sensitivity is improved during smooth pursuit, and also the prediction of moving targets, the direction of moving targets. So these results demonstrate that the movement of our eyes and visual information uptake are really intricately intertwined. And the two processes interact to enable an optimal vision of the world. And I think we can only investigate this optimal vision by allowing the eyes to move and not by having this small fixation spot. Thank you very much. And these are all the people who did the work.